So we'll start out by talking about physical controls on microbial growth. The first one we'll cover is heat. You can use heat to either disinfect or sterilize a sample or an object. And as we've discussed, heat affects enzymes in the plasma membrane. So we talked about the reasons for that in the microbial growth series when we talked about temperature ranges that various microbes can tolerate. So we talked about how hot and cold has an effect on enzymes and how hot and cold has an effect on the plasma membrane. There's two styles of heat. You can have moist or dry. Moist is more effective. So moist heat would be steam or boiling water, and this physically disrupts the cellular molecules. So this is going to be an example of disinfection or sterilization. Water moves heat around quickly and evenly is the reason, because water is a really good conductor of heat, meaning it can carry heat from one place to another. You can tell that water is a good conductor of heat because if you sit in water that is 70 degrees Fahrenheit for long enough, you will eventually get hypothermia. 70-degree Fahrenheit water feels cool because heat is moving from your body to the water very quickly, and yet 70 degrees Fahrenheit air temperature feels perfectly comfortable. You're not going to get hypothermia from 70 degrees Fahrenheit air. And that's because air is not a good conductor of heat. It doesn't pick up the heat from your body the way that water does. So heat, just like we talked about molecules move from high to low concentration, heat also moves from high to low. So if your body is warmer than the outside environment, then heat is going to move from your body to the outside environment, but it just moves faster in water. It moves out of your body. So if you're in 70-degree Fahrenheit water, it's moving out from your body faster than the same temperature air, and it'll actually move out faster than you can regenerate that heat. So I said that wet heat can disinfect or sterilize. Well, without an autoclave, it's just disinfecting. Endospores are resistant to wet heat. So an autoclave is shown here. An autoclave combines heat with pressure to sterilize. So if you use wet heat in conjunction with pressure, then you can sterilize and get rid of even endospores. And you have autoclaves in laboratories and in hospitals to sterilize equipment. All right, let's talk about dry heat. Dry heat is like burning something to ashes or oxidation, and it can be used to sterilize if you make something hot enough for long enough. But it really requires a lot more time to damage cells relative to wet heat. And that's because air is like we just talked about, it's a poor conductor of heat. You can intuit that dry heat is less damaging because air is a poor conductor of heat. By comparing what it would feel like to put your hand in boiling water versus holding it in the air of a 212-degree Fahrenheit oven. Both temperatures are the same, but your hand doesn't get burned by holding it in the air of the oven. Now, if you touch the metal of the oven, that's its lowest setting, right? I mean, you might not even have a setting that low in your oven. So 200-degree Fahrenheit oven, that's its lowest setting. You can stick your hand in there and it doesn't, you can feel it's warm, but it doesn't hurt you unless you touch the metal. Metal is a good conductor of heat as well, but the air is not. But if you put your hand in the water while it was boiling, that's actually the same temperature as the air inside that oven, but the water is such a better conductor of heat that it puts that heat straight into your skin and damages your skin, right? So dry heat works by oxidizing molecules. Think about keeping paper in an oven at about 200 degrees Fahrenheit. If you leave it there long enough, it will get brown and charred, even though this is only half the temperature required to burn paper. The heat is helping oxygen and the carbohydrates in the paper to reach their activation energy so that they react. Because oxygen is moving around so much in the hotter air, it's colliding at higher speeds and at higher frequency with the paper. So remember collision theory. So when you burn paper, it's the same thing as when you metabolize carbohydrates in your body. We talked about metabolism already. It's just that in your body, each step of those chemical reactions is separated, taken apart and done in slow motion, or not in slow motion, but done in series. It doesn't all happen. The reaction doesn't all happen at once. In an oven, it's all happening at once, but you can get that paper to burn those calories from that paper just by putting it in a hot oven, but it just takes a long time with that low a temperature. Pasteurization. Pasteurization is just enough heat to reduce microbial numbers. This is not sterilization. And you use this when you don't want to ruin the nutrients and flavors of, like, for a food, for example. We use pasteurization for milk, we use it for wine. Commercial sterilization is more intense than pasteurization, but still its only purpose is to kill Clostridium difficile. Anything that can survive higher temperatures than Clostridium difficile endospores is generally not a human pathogen, so it's usually just fine for commercial sterilization is usually just fine, even if there are some species of microbes left over. Filtration. You could consider this a form of sterilization. It's actually probably more accurately called degerming, but we tend to, in laboratories, we tend to say that this is a form of sterilization. So filtration is when you run water or air through a filter, and a filter is a membrane with very, very tiny pores. And if those pores are small enough, they will catch everything moving through them, so that on the other side, the air or water that's moved through to the other side no longer has any microbes in it, no viruses, no prions, nothing. So this is just about the size of the pores in that membrane. So if you're putting water through a filter, like this guy who's camping here, if the pore size is small enough, it will capture anything that's in that water so that the water is pure on the other side. So this is a way to sterilize things that are heat sensitive, such as vaccines or media with nutrients that would degrade under intense heat. Though you don't buy hospital filters at REI, right? So, but those filters that you can buy at REI for camping, they're good for taking out anything that would make you sick in the water. So it's often considered a method of sterilization because no microbes come through the other side, but actually more accurately it would be degerming, because you're not killing the microbes, you're removing them. So you could call it either, but most people would say it's sterilizing because the product coming out the other side is microbe free. All right, cold. Cold is a method of slowing microbial growth, so we call that bacteriostasis. And for this, again, you have to remember collision theory. So as you remove thermal energy from water or air, what happens is the molecules slow down. And they slow down, when you get to ice, the water molecules are really not moving at all. And so if you freeze a living thing, all the molecules stop moving, things become solid that weren't. And everything has slowed down so much that all the biochemical reactions involved in metabolism, or like building new cell parts, repair, digestion, reproduction, all of the chemical reactions responsible for those things have slowed way, way down, and maybe even stopped completely. Okay. Now, so when you put milk in your refrigerator, it will go bad eventually, but it'll take a lot longer than it would have if you left it on the counter. And that's just because the microbes in it they might be metabolizing the milk still, but at a much, much slower rate. Now, water expands when it settles as ice. This is a special property of water. Most things take up more volume as a gas or liquid relative to their solid state. So the solid state takes up the least volume, but it's the opposite for water. So water expands as it freezes. So it actually takes up more volume as it freezes. And this can cause some cells to swell and burst when they turn into ice, when they freeze, so that when they thaw, this makes them no longer viable because the cell membrane has fractured. But even though that happens to some cells, freezing is not considered a form of sterilization because so many bacteria can survive it. It just freezes them by halting all the chemical reactions within them, but many of them can come back once they've been thawed. Not all of them, but enough. So when we freeze something, we're halting microbial growth, but we don't assume that the microbes have been killed when we thaw the item again. All right, pressure. Pressure alters the structure of important molecules. It puts a lot of pressure, pressure will put a lot of strain on molecular bonds, causing them to break. It can put a lot of strain on the structures of the cell, causing the cell structure to break. Maybe even the cell wall will break. Maybe the cytoskeleton will break. Maybe the cell membrane will burst. Okay, it's sort of like squishing a balloon as hard as you can until it pops. This is a means of disinfection. There are endospores that are resistant to pressure. So remember though, an autoclave combines pressure and moist heat, and the two together can be used to sterilize. Desiccation. Desiccation means to dry something out. This is, again, a form of bacteriostasis, sort of like freezing. It halts metabolism. And to understand this, you again have to remember collision theory. So for molecules to react, like in biochemical reactions, metabolic reactions, they have to come in contact with each other. They have to collide. These molecules are typically floating in water. The cell is mostly water, right? So water allows most molecules dissolve in water. This allows molecules to move around. This allows them to come in contact with each other, allowing enzymes to encounter substrates and convert them into products. If you remove the water, you prevent any reactions from happening within the cell because everything now is just sitting on a solid surface. So if you think about it, like think about your room, you've got, I've got some shoes on the floor over there and a blanket on the floor over there, and they're not colliding with each other because they're sitting on a dry, solid surface. Now, if I filled the room with water, everything would start floating around, and some of those items would start colliding with each other, right? So if you need collisions to have a chemical reaction, collisions between reactants, between substrates and enzymes, if you take away the water, that's not going to happen. Everything's just going to sit still on a solid surface. So for this reason, most bacteria just cannot grow in dried foods very well. That's why dried foods last for so long. Think about powdered milk or flour or even sugar in your, in your cupboard. These keep for a long time, even though they are filled with nutrients that microbes want. But you don't see any microbial growth on them unless you soak them in water. If you did, you'd see growth very quickly. If here's some dried milk, some powdered milk, that'll keep in your cupboard forever, essentially, because it's so dry. That halts microbial growth completely. But if you were to add water to that and leave it in your cupboard, it would be bad in a day, right? Because it is filled with nutrients that microbes love. They're just not growing there because it's too dry. So that's another way to control microbial growth is desiccation. Osmotic pressure is very similar to desiccation. So you can control microbial growth through osmotic pressure. So osmotic pressure, again, that has to do with pressure due to water movement. And actually here we're going to have negative osmotic pressure. We're not going to fill the cell to bursting. Here, to control microbial growth, we are going to suck the water out of it by adding lots of salts or sugars to the microbial environment, by making the outside environment hypertonic, or more solutes relative to the inside of the cell, of the microbial cell. This effectively then, this is desiccation. You're drying out the cell because if you make the outside environment hypertonic to the inside of the bacteria, water is going to move out. Water moves down its own concentration gradient from where it is most pure to where it is less pure. So if there's more solute outside than inside, the water is going to move out into the solute. And that's going to dry out the inside of the cell. So that's a lot like desiccation. It basically does the same thing. So that's, again, going to be biostasis, or slowing or halting microbial metabolism and growth. So we do this for pickling. To pickle something, you just put it in a lot of salt water and it doesn't go bad, does it? That jar of pickles can be in your cupboard for ages and it won't go bad. Same with making jam. It's filled with sugar, right? It seems like the bacteria would like that, but it is simply too hypertonic to the internal bacterial environment. It's not going to grow, or not going to grow very fast, right? I mean, jam will go bad eventually, but not nearly as fast as it seems like it should. And that's just because it's hypertonic because of all of those sugars dissolved in it relative to the inside of the bacterial cell. All right, soaps. This is a form of degerming. So degerming is just removing bacteria from an area. And actually a lot more, it doesn't seem like it's not a very harsh treatment, so you might not think it's effective, but it actually is quite effective. So hand washing, if you wash your hands well, you can remove quite a number of microbes from the surface of your hands. So soaps are amphipathic molecules, meaning they have a hydrophilic end and a hydrophobic end. The hydrophobic end mixes with all the oils on your skin, and the hydrophilic end mixes with water, so you can rinse them off. So oils, usually it's hard to clean with water, right? If you think about the oils, if you cook some bacon in a pan, that pan, you're not really going to be able to clean it with just water, right? Not very easily. You're not going to get all the grease off. But if you add a little soap, it cleans much more easily. And that's because the soap can mix both with the oils and with water, so that makes the water more effective at rinsing. So that's not killing microbes, that's just removing them from a surface. All right, let's talk about radiation. So here you have the light spectrum. So this is the band of visible light that we can see. This is what's visible to us. Different animals can see different wavelengths of light, but this is what's visible to us. So there's this narrow band, but then there are wavelengths of light on either side of this, right? There's non-ionizing radiation, and then there's ionizing radiation. So over on this side, so red, if you're talking about just the visible light spectrum, red has the least energetic wavelength. So the wavelength is wide, so it's a long wavelength, and it's also low energy. Purple has the highest wavelength, the fastest wavelength, and the highest energy wavelength of the visible light spectrum. So purple is more energetic than red. And so over here you have, on this side of purple, you have ultraviolet, so it's like red, orange, yellow, green, blue, violet. And then ultraviolet, we can't see ultraviolet, so that's just in the spectrum that we can't see, but it's even more energetic than purple. Okay, and then there's X-rays after that and gamma rays after that. So the farther you go to the right of this graph, the more energetic those wavelengths are, and the further you get to the left, then you get infrared, which we can't see. That's like heat. So some animals can see infrared spectrum, like snakes, so they can see warmth. For their mammal prey. And then even slower wavelengths than that, you have microwaves, radio waves, et cetera, et cetera. So the further you get to the left, the lower the energy. So ionizing radiation is everything to the right of the light spectrum, and non-ionizing radiation is light spectrum and everything to the left of that. So ionizing radiation includes X-rays, gamma rays, electron beams. The main effect is creating hydroxyl radicals from water. So if you direct an ionizing radiation beam at something, the main way it's killing the thing or damaging the thing is by acting on the water. So the water gets split into hydroxyl radicals, which is a reactive oxygen species, which we just talked about. So you're creating these reactive oxygen species that are toxic and that damage the cell. And then non-ionizing radiation, that actually does include some UV light. So some UV light is ionizing, other UV light is non-ionizing. So it's split. So you have the slowest wavelengths of ultraviolet light are non-ionizing, and then visible light and everything to the left of that. So the only sterilizing wavelength for non-ionizing is that little bit of ultraviolet light. So ultraviolet light you can use for sterilizing. With a sufficient length of time and proper conditions, making sure, for example, that UV light can penetrate the substance. Both can be used as sterilizers, both ionizing and non-ionizing. And they are often used in labs and hospitals to help sterilize equipment and solutions that cannot be heated without destroying them. So you can use ultraviolet light, whether the ultraviolet is ionizing or non-ionizing, and you can use any kind of ionizing radiation to sterilize. So what the ultraviolet does is it damages DNA. So it's not just through hydroxyl radicals. It also, if the ultraviolet light hits DNA, it'll damage the DNA, which can kill an organism. So both Ultraviolet light is sterilizing, and all the ionizing is sterilizing. But you have to, like I said, you have to make sure that the radiation can penetrate all parts of the sample. So if there's anything that didn't get hit by the ultraviolet light, like if you've got this really murky sample and the radiation can't penetrate, there's going to be pockets where things didn't get killed. So you want to make sure that all components are being hit by the radiation for a sufficient length of time. So in laboratories, at laboratory benches where you do microbiology, they often have UV lights. And so what you can do is you can soak it down with alcohol, turn on the UV light, walk away for half an hour, and then come back, and your station should be sterilized. Microwaves are not very powerful at all. They can kill microbes only in as much as they can boil water. But microwaves are full of microbes. Microwaves in your kitchen, they are disgusting. And drier foods like meat are hard to cook safely because of unequal heating throughout. So you cannot kill a thing with microwaves directly. You can boil water with microwaves, and the boiling water, we talked about how moist heat can be a disinfectant, right? So as long as water is being boiled thoroughly, you can kill microbes in a microwave, but it's through the boiled water, not through the microwaves themselves.